All right, let's go.
Number 15. Your brain is 0.5% plastic, and that number is rising. In February 2025, a study published in Nature Medicine by researchers at the University of New Mexico examined brain tissue samples of 91 human cadavers. They were looking for microplastics. They found them everywhere. The median concentration of microplastics in brain tissue from samples collected in 2024 was nearly 5,000 micrograms of plastic per gram of brain tissue. That is almost 0.5% of your brain by weight, not trace amounts. 0.5% plastic sitting in your frontal cortex. It gets worse. When the team compared 2024 samples against samples from 2016, the concentration had increased by approximately 50% in 8 years. More plastic in brains collected recently than in brains collected 8 years ago. Lead author Matthew Campen described the result as unbelievable and stated, "You can draw a line. It's increasing over time. It's consistent with what you're seeing in the environment." The brain accumulates more microplastic than any other organ, more than the liver, more than the kidneys, 20 times more than surrounding tissue in some samples. The predominant polymer found was polyethylene, the same plastic used in shopping bags, food wrapping, and bottles. Nanoparticles small enough to cross the blood-brain barrier accumulate preferentially in brain tissue. The brains of patients who died with dementia contained significantly higher concentrations than the brains of cognitively healthy individuals. Whether this relationship is causal, whether plastic in the brain is contributing to cognitive decline, is not yet confirmed. What is confirmed is that your brain contains plastic, that the concentration increases every year, and that there is currently no known mechanism to remove it. Number 14. A parasite in 3 billion human brains is altering behavior, and most people do not know they have it. Toxoplasma gondii is a single-celled parasite. Its primary host is the domestic cat. It reproduces sexually only inside cat intestines. To get back to a cat, it needs to travel through an intermediate host, typically rodents. So it evolved a mechanism to make that journey more likely. It makes infected rodents lose their fear of cats, specifically. Not all threat responses, just the one for cats. Infected mice approach cats' scent rather than avoiding it. They seek out their predator. They get eaten. The parasite completes its life cycle. Toxoplasma achieves this by forming cysts in the brain, primarily in the amygdala, the region responsible for fear and threat processing, and altering the neural chemistry of its host. In rodents, this effect is well documented and robust. In humans, the picture is more disturbing. Approximately 40% of the global human population is estimated to carry a latent toxoplasma infection. In some parts of the world, the prevalence reaches 70%. Most of these people have no symptoms. The infection is chronic, lifelong, and for most people, entirely asymptomatic. But it is not entirely without effect. Studies led by Jaroslav Fleg at Jaroslav University in Prague over decades found that toxoplasma-infected humans show measurable changes in personality and risk behavior. Infected men show higher risk-taking, increased impulsivity, slower reaction times, higher rates of car accidents, statistically confirmed across multiple countries. Infected individuals show subtle alterations in testosterone levels. Some studies have found correlations with toxoplasma infection and higher rates of schizophrenia. Specifically, the parasite's metabolic byproduct near dopamine synthesis pathways in the brain may sensitize dopamine signaling in ways that parallel schizophrenic neurochemistry. A parasite that evolved to manipulate rodent brains for its own reproductive benefit is sitting in the brains of 3 billion humans, altering their behavior, subtly, persistently, in ways that look, from the outside, like personality. Number 13. Every memory you have is being rewritten every time you remember it. Memory feels like playback, like accessing a stored file, retrieving something fixed. That is not what happens. Every time you recall a memory, it becomes temporarily unstable. The neurons that store it, the engram cells holding the pattern, reactivate. And in that reactivated state, the memory is vulnerable. It can be altered, strengthened, weakened, updated with new information, contaminated by current context, or partially replaced by something that did not happen. This process is called reconsolidation. Every recall is a partial rewrite. The memory you access after remembering something is not the same memory you had before you remembered it. It has been reconstructed, subtly modified, resaved. Elizabeth Loftus at the University of California demonstrated the fragility of this system repeatedly over decades. In one famous experiment, participants watched footage of a car crash. Some were later asked, how fast were the cars going when they hit each other? Others were asked, how fast were the cars going when they smashed into each other? The word smashed produced significantly higher speed estimates. And when participants were asked a week later whether they had seen broken glass in the video, there was no broken glass. The smashed group reported seeing it twice as often as the hit group. A single word changed what they remembered seeing. In more recent research, entire fabricated childhood memories have been implanted in laboratory subjects using a technique called guided imagery combined with false familiarity. Study participants came to believe they had been lost in a shopping mall as a child, or had a frightening encounter with a vicious animal, or had witnessed a strange event, none of which happened. They reported these memories with emotional conviction. They added sensory details. They were indistinguishable from the inside from real memories. In a 2025 study examining how AI-edited images and videos affect recall, researchers found that showing participants digitally manipulated photographs caused them to remember events that never occurred with the same confidence as their real memories. Your most vivid memories are not records of what happened. They are reconstructions made slightly imperfect every time you access them. The version of your past you carry in your head has been modified thousands of times. You will never know by how much. Number 12. Your brain made your decision before you knew you were deciding. In 1983, neuroscientist Benjamin Libet ran an experiment. He asked participants to flex their wrists whenever they felt like it while monitoring their brain activity. The participants reported the exact moment they felt the urge to move by watching a rapidly rotating clock. The brain began showing activity, what he called the readiness potential, up to half a second before participants reported consciously deciding to move. The implication was already uncomfortable, but it got significantly worse in 2008. John Dylan Haynes at the Max Planck Institute in Leipzig used fMRI, which captures brain activity across regions rather than just local electrical signals, and found that brain activity in the frontopolar cortex and parietal cortex could predict which button a participant would press up to 7 seconds before the participant reported consciously making the decision. 7 seconds. The brain had already made the choice 7 seconds before the person experienced choosing. Further experiments tested whether this extended to abstract cognitive decisions. Participants freely chose whether to add or subtract two numbers. Brain patterns indicating the chosen operation became detectable up to 4 seconds before conscious awareness. The decision happened first. The experience of deciding happened after. What you experience as the moment of choice may be the brain's report to consciousness of something already determined, not the cause, the announcement. This does not end the free will debate. The Sugar accumulator model, proposed in 2012, offers an alternative reading. The readiness potential may reflect stochastic neural noise building to a threshold rather than a predetermined decision. The debate is alive, but the original finding has been replicated. Your brain acts before you know it has decided to act, every time, without exception. Number 11. Your conscious experience is lagging 500 milliseconds behind reality. Right now, as you listen to this, you are not experiencing the present. You are experiencing 500 milliseconds ago. Neural signals from your sensory organs, eyes, ears, skin, take time to reach the brain, travel through processing pathways, and integrate into conscious experience. The brain then performs a process called post-diction. It takes the completed, processed sensory data and back projects it in time, making you feel as though you experienced the event at the moment it occurred, rather than half a second afterward. This is not lag you can feel. The brain conceals it. It stitches together a seamless, real-time feeling experience from information that is continuously delayed. When you catch a ball you consciously experienced as catching in real time, you were actually processing information from half a second earlier. When you hear a loud sound and flinch, you are responding to an event that your conscious self has not yet experienced at the moment your body begins to react. In 2025, a team of neuroscientists discovered what they called a reality signal, a specific neural signature generated by a brain region that distinguishes between imagined experiences and real perception. When you imagine an apple, the brain activity is similar to when you actually see one. The reality signal is what tells your brain the difference. It is not a property of the incoming sensory signal itself. It is generated by the brain, applied to perception, and used to label some experiences as real, which means what you call reality is not something arriving from outside. It is a category your brain assigns to its own internal activity. It can be wrong. It is wrong in hallucinations, in vivid dreams, in some psychotic episodes, and potentially in scenarios no one has yet thought to test. Number 10. The brain produces pain in places where nothing is happening. Chronic pain is not damage, not inflammation, not injury. In millions of people, it is the brain producing a pain signal with no corresponding threat in the body. Phantom limb pain is the textbook example. Amputees experience intense, often burning pain in limbs they no longer have, sometimes for decades. The limb does not exist. The pain is entirely real. It is produced by a brain that has a map of the body called the somatosensory cortex, where representations of the missing limb still exist and remain active. The pain signal comes from the map, not from the territory. But phantom limb pain is simply the clearest example of a general principle that neuroscience has been establishing for decades. Pain is not a signal arriving from the body to the brain. Pain is an output signal the brain generates in response to its interpretation of the body's state. Sometimes that interpretation is wrong. Sometimes it becomes self-sustaining, a feedback loop with no peripheral input required to keep it running. Fibromyalgia, complex regional pain syndrome, and some forms of lower back pain appear to involve central sensitization, a state where the pain-processing regions of the brain become hypersensitive. They fire pain signals in response to stimuli that would not normally be painful, to light touch, to temperature changes, to emotional stress, not because the tissue is damaged, because the brain's threat evaluation system has been recalibrated. The WHO estimates over 1.5 billion people worldwide live with chronic pain. A significant proportion of them may be experiencing pain produced entirely by a brain that has lost its calibration, generating suffering that has no material cause and responds poorly to treatments designed for physical injury. Number 9. Your brain is actively hallucinating your entire reality all the time. Neuroscientist Anil Seth at the University of Sussex has argued, based on decades of research and predictive processing, that conscious experience is a controlled hallucination, not metaphorically, mechanically. The brain does not receive sensory data and process it into a picture of the world. It runs a continuous prediction of what it expects the world to be, generates an internal model, and uses incoming sensory signals primarily as error correction signals, inputs that update the model when it deviates too far from prediction. The vast majority of what you see, hear, and feel is generated by the model, not transmitted from the outside world. You are not experiencing the room you are in right now. You are experiencing your brain's current best guess of what the room looks like, continuously corrected by light hitting your retinas. This is not a philosophical abstraction. It has direct, measurable consequences. The rubber hand illusion demonstrates it. If you watch a fake rubber hand being touched while your own hidden hand is touched in synchrony, your brain reassigns ownership of the rubber hand. You feel the touch in the rubber. When the rubber hand is threatened, your skin generates a stress response, detectable in galvanic skin measurements, as though the threat is real. The brain's model incorporated the rubber hand, and it defends it. Hallucinations in psychosis are not the brain receiving false input from the outside. They are the brain's internal, predictive model running without adequate correction from sensory reality. The model generates sensory experience, voices, visions, and because the error correction mechanism is impaired, the experience is indistinguishable from genuine perception. What keeps your hallucination calibrated is continuous sensory input. Remove that input through sensory deprivation, and the hallucinations begin within hours for most people, not because something is added, because the correction signal is removed and the model runs unchecked. Number 8. Herpes simplex is in most human brains, and it may be triggering Alzheimer's. Herpes simplex virus type 1, HSV1, is carried by approximately 67% of the global population under the age of 50. Most people know it as the virus that causes cold sores. What most people do not know is that it takes up permanent residence in the nervous system. After initial infection, HSV1 retreats to the trigeminal nerve ganglia, nerve clusters at the base of the skull, and enters a latent state. It stays there for life. It reactivates periodically during immune stress, illness, fever, sunburn, emotional stress, travels back along the nerve to the skin and produces a cold sore, then retreats again. Some reactivations travel in the other direction, into the brain. In 2018, researchers analyzing post-mortem brain tissue from over 600 individuals found that people with higher HSV1 viral load in the brain had higher levels of amyloid beta and tau protein, the two hallmark pathological features of Alzheimer's disease. The virus appeared to trigger the formation of amyloid plaques as an immune response. The plaques, classically understood as the cause of Alzheimer's, may actually be the brain's antiviral defense mechanism, encasing the virus and protein to contain it. A 2019 study in Taiwan that examined the health records of over 33,000 people found that individuals with an HSV infection severe enough to be documented medically had a 2.56-fold increased risk of developing dementia. Crucially, those who received antiviral treatment showed dramatically reduced dementia risk. The same risk reduction in subsequent years was between 89 and 96%. As of 2026, Alzheimer's disease research is at an inflection point. The amyloid hypothesis, plaques cause Alzheimer's, has failed to produce effective treatments in trial after trial. In June 2026, a study published in Nature proposed that amyloid beta does not directly cause neurodegeneration, but instead disrupts the tau protein, which then damages neurons. The underlying trigger driving the entire cascade may be chronic viral infection in the brain, not a protein misfolding disorder, an infectious disease that takes decades to kill. If that is confirmed, a vaccine against HSV1 or early antiviral treatment could prevent the most common form of dementia, and a virus already inside the brains of most people reading this is the prime suspect. Number 7. Chronic stress physically shrinks the brain. Not metaphorically, not gradually and barely detectably, measurably, permanently, in structures critical to memory and emotional regulation. The hippocampus, the region responsible for forming new long-term memories and spatial navigation, contains receptors for cortisol, the primary stress hormone. During normal stress responses, cortisol spikes briefly and returns to baseline. During chronic stress, cortisol remains chronically elevated. And chronically elevated cortisol triggers apoptosis, programmed death in hippocampal neurons. Studies in both humans and animals have consistently documented hippocampal volume reduction in individuals with chronic stress, PTSD, and major depressive disorder. The reduction is not subtle. Adults with severe, prolonged depression show hippocampal volumes significantly smaller than healthy controls. Individuals with PTSD show similar structural changes. The longer the stress exposure, the greater the loss. The amygdala, the threat processing region that drives fear and anxiety responses, responds to chronic stress in the opposite direction. It grows. It becomes hyper-responsive. The already overwhelming anxiety of chronic stress actually rewires the threat detection system to be more sensitive, not less, more likely to detect danger, more likely to generate the cortisol that shrinks the hippocampus further. This is a feedback loop with no natural braking mechanism under sustained stress conditions. The brain under chronic stress becomes structurally worse at regulating its own stress response over time. A 2025 study found that some hippocampal volume can recover with successful antidepressant treatment and stress reduction. The brain retains some plasticity, but the recovery is partial. Some of what chronic stress takes, it does not give back. Number 6. Your brain cannot tell the difference between physical and social pain. The neural substrates of physical pain and social rejection overlap almost completely. In a landmark study by Naomi Eisenberger and Matthew Lieberman at UCLA, participants were scanned using fMRI while playing a virtual ball-tossing game, Cyberball, in which they were eventually excluded from the game by the other players. Being socially excluded activated the dorsal anterior cingulate cortex and the anterior insula, the same regions activated when you stub your toe or burn your hand. Social rejection causes activity in the pain matrix, not a metaphor of pain, not pain-like discomfort, the same neural regions, the same circuits. That same study found that individuals who scored higher on self-reported emotional reactivity to rejection showed stronger pain matrix activation during exclusion. That higher activation correlated with higher scores on measures of everyday social distress. Acetaminophen, paracetamol, the over-the-counter painkiller, has been shown in double-blind trials to reduce self-reported hurt feelings following social exclusion, and to reduce the neural pain response to social rejection and fMRI imaging. A headache tablet working on a broken heart. Not because social pain is less real than physical pain, because they are the same pain. This finding has implications that extend well beyond psychology. Social isolation activates the same threat systems as physical injury. The brain treats loneliness as a survival threat. Long-term social isolation produces neurological changes measurable in imaging studies, changes in white matter, in cortical thickness, in default mode network connectivity, that overlap with the changes produced by chronic physical stress. Number 5. The glymphatic system cleans your brain while you sleep, and when it fails, you get Alzheimer's. In 2013, researchers at the University of Rochester discovered that the brain has its own waste clearance system, one that had been completely unknown to science until then. The glymphatic system, a network of channels surrounding blood vessels in the brain, through which cerebrospinal fluid flows, but only during sleep. During waking hours, glymphatic flow is nearly zero. During sleep, specifically deep, slow-wave sleep, the interstitial space between brain cells expands by approximately 60%. Cerebrospinal fluid rushes through these channels and flushes out metabolic waste products, including amyloid beta and tau, the proteins that accumulate into the plaques and tangles of Alzheimer's disease. Your brain accumulates these waste products continuously while you are awake, doing the metabolic work of thinking. Sleep is when they get cleaned out, every night, as long as the glymphatic system functions correctly. When sleep is disrupted chronically, repeatedly, waste accumulates faster than it is cleared. A single night of sleep deprivation produces measurable increases in amyloid beta in the cerebrospinal fluid. 2 weeks of sleeping 6 hours per night produces cognitive deficits equivalent to 24 hours of total sleep deprivation. Chronic sleep restriction over years increases Alzheimer's risk measurably. The system works in a specific sequence. Deep sleep first. The initial phases of the sleep cycle drive the strongest glymphatic flow. Alcohol disrupts deep sleep architecture, so does ambient light and the blue light from screens specifically. All of them reducing the phase of sleep when the cleaning happens. The brain generates its own toxins. It cleans them up at night. Skip the cleaning long enough and they accumulate into disease. The mechanism for one of the most feared diseases in human aging is, in part, the consequence of not sleeping. Number 4. The default mode network runs constantly when you are doing nothing, and it is linked to depression, rumination, and mental illness. When you are not focused on a specific task, the brain does not rest. A network of regions, the medial prefrontal cortex, the posterior cingulate cortex, the angular gyrus, the hippocampal formation, activates together. This is the default mode network, or DMN, first described in 2001 by Marcus Reichel at Washington University, initially assumed to be background noise, now understood to be one of the most active and energetically expensive systems in the entire brain. The DMN generates self-referential thought, mental simulation of future scenarios, replay of past events, social cognition, modeling other people's minds, the narrative of your identity, the sense of being you. It also generates rumination. Studies consistently show that individuals with major depressive disorder have hyperactive default mode networks, excessive connectivity between DMN regions, a system that runs too hard, that loops back on itself, that generates self-critical, pessimistic, repetitive internal narratives that the person experiencing them cannot stop. The DMN is also hyperactivated in anxiety disorders, in PTSD, where it replays traumatic experiences, in obsessive-compulsive disorder, where it generates intrusive thoughts the person knows are irrational but cannot dismiss. Meditation as a practice systematically reduces DMN activity. fMRI studies show that experienced meditators have reduced connectivity in the DMN specifically, the same regions quieter. Antidepressants in successfully treated patients normalize DMN hyperactivation. Ketamine, currently the most rapidly acting antidepressant available, produces its effects partly through rapid disruption of pathologically hyperconnected DMN circuits. In some studies, a single dose produced measurable reductions in DMN connectivity within hours. The part of the brain you use when you think about yourself is the same part that produces depression when it runs too hard. The narrative voice inside your head is the most expensive metabolic activity your brain performs, and in many people, it is also the most destructive. Number 3. Adolescent brains do not finish developing until age 25, and decisions made before then happen in a fundamentally different brain. The prefrontal cortex, the region responsible for impulse control, risk evaluation, long-term planning, and the ability to consider future consequences, is the last part of the brain to complete development. It finishes myelinating, the process by which neural connections become faster, more stable, and more reliable around age 25, not 18, not 21, 25. Before that age, the prefrontal cortex functions less efficiently. Connectivity with the limbic system, the emotional, reward-driven, impulsive regions, is less regulated. The balance of influence between immediate reward and long-term consequence is structurally tilted toward the immediate. This is not a failure of moral development or willpower in young people. It is the literal state of the organ. The hardware responsible for weighing consequences is not finished being built. Legal systems in many countries set the age of criminal majority at 18, the age of alcohol consumption at 18 or 21, the age of military service at 17 or 18, all below the age at which the region of the brain responsible for assessing risk and long-term consequences finishes construction. The neuroscience of adolescent brain development has been used in U.S. Supreme Court cases, including Roper v. Simmons in 2005 and Miller v. Alabama in 2012, to argue against mandatory life sentences for crimes committed by individuals under 18, precisely because the brain doing the decision-making was structurally different from an adult brain. The Supreme Court accepted this argument both times. Decisions made before 25, whether to try a drug, to commit to a relationship, to choose an educational path, to engage in a dangerous behavior, happen in a brain whose risk evaluation hardware has not been fully installed. Number 2. Your brain has a finite attention budget, and almost everything in the modern world is engineered to steal it. The brain's attentional resources are finite, neurologically fixed. The anterior cingulate cortex and the prefrontal cortex act as an executive attention system, directing what gets processed consciously, what gets filtered, and what gets access to working memory. This system operates on a limited energy budget and fatigues under sustained load. Directed attention fatigue, studied extensively by Stephen and Rachel Kaplan at the University of Michigan, occurs when the involuntary attention system is overwhelmed and the directed attention system is chronically recruited to compensate. The cognitive load of urban environments, screen-based work, constant notification interruptions, and the algorithmic design of social media platforms optimized specifically to prevent attentional disengagement produces measurable depletion of the directed attention system over time. In 2024, a large longitudinal study tracking cognitive performance across 90,000 participants found that heavy social media users showed measurable reductions in sustained attention capacity over 5 years. The ability to hold focus on a single task without disengagement degraded significantly in individuals whose digital environment was designed to prevent that focus. The attention economy, the business model of platforms that sell your attention to advertisers, operates by exploiting specific features of neural architecture: variable reward schedules, social validation signals, infinite scroll. These features target the dopaminergic reward circuitry of the striatum and the amygdala's threat monitoring function simultaneously. They keep the brain in a state of low-grade vigilance, scanning for the next piece of potentially important social information that prevents the sustained directed attention the frontal cortex uses for deep cognitive work. The prefrontal cortex, the thing that builds your capacity for complex thought, long-term planning, and creative problem-solving, competes with systems that evolution and algorithm design have both made extremely effective at grabbing control. Most of the time, the algorithm wins. Number 1. Your brain is filling in most of what you think you are seeing. The visual field appears seamless, continuous, detailed from edge to edge. None of that is true. Your eyes have a blind spot, the point where the optic nerve exits the retina, which contains no photoreceptors and is therefore completely blind. Your brain fills it in so seamlessly you have never noticed it in your life. You can find it only by closing one eye and moving a small object across your visual field until it disappears. Your peripheral vision has almost no color resolution, almost no fine detail. The region of sharp, full-color, high-resolution vision, the fovea, covers approximately 2 degrees of arc, about the width of your thumbnail at arm's length. Everything outside that area is low-resolution, low-color information that the brain upsamples, fills in with guesses, to produce the impression of a rich, detailed, uniformly clear visual field. Your eyes make rapid, involuntary movements called saccades 3 to 4 times per second. During each saccade, vision is effectively suppressed to prevent the smearing blur that would otherwise be visible. Your brain bridges these suppression periods with seamless, fabricated continuity. Your retinal image is two-dimensional. Your experience of depth, distance, and three-dimensional space is constructed by the brain using binocular disparity, motion parallax, texture gradients, and learned associations. None of it arrives pre-assembled from the world. What you experience as vision is a high-confidence fabrication, the brain's best guess at what the world in front of you contains, based on a tiny high-resolution window, a large low-resolution periphery, and constant interruptions it smooths over before you notice them. The confidence with which you experience this fabrication as reality is indistinguishable from the inside from the confidence you feel about things that are actually real. The brain does not show you the world. It shows you its model of the world, and it does not tell you that is what it is doing. 15 things scientists have found about the human brain that should fundamentally change how you think about what you are. Your brain is currently 0.5% plastic by weight and rising. A parasite sits in the brains of 3 billion people, altering their behavior. Every memory you have has been modified each time you remembered it. Your brain made every decision you think you made before you knew you were making it. Your conscious experience is running 500 milliseconds behind reality. Your brain produces pain in places that don't exist. Everything you see is a high-confidence hallucination. A common virus in most brains may be the true cause of Alzheimer's. Chronic stress physically deletes brain tissue. Social rejection activates the same circuits as physical pain. Sleep cleans the brain, and skipping it accumulates the proteins of dementia. The voice inside your head that narrates your life is also the primary mechanism of depression. Your frontal cortex was not finished being built until you were 25. Your attention is being systematically extracted by systems designed around your own neurology. And the seamless, detailed, three-dimensional reality you are experiencing right now is a fabrication your brain produces from two small, constantly interrupted patches of high-resolution data and a lot of educated guessing. This is the organ producing your experience of everything, including this sentence, including your response to it. Subscribe to Cosmic Hives. See you in the next one.